Hi everyone. I'm really delighted to give you today's lecture. This is really the Myron Gordon lecture that was supposed to be held in Japan this year, and very disappointingly, I will have to bring this to you from Chicago, and you won't even see much of the environment, but I hope nonetheless you'll enjoy what I have to say. I decided to speak to you about our work that relates to regulatory T cells because regulatory T cells impact both autoimmune and anti-tumor responses. And so it kind of comes into the interplay that I'm very interested in, namely the understanding of what happens in vitiligo and what happens in melanoma. So I'm Caroline Poole. I'm a full professor of dermatology at Northwestern University in Chicago. I also have a secondary appointment in microbiology and immunology. So today's prize is all about Myron Gordon. He was somebody who passed away, unfortunately, before I was born, so I never had a chance to meet him, but he was a zoologist and geneticist, and the model he studied mostly was Xiphophorus, and he did this to study melanoma biology, did most of his work in New York. And helped us understand genetic factors and heredity as it is involved in melanoma formation. And that's some groundbreaking work that he did in this regard. He also organized the four first IPCC meetings. So I think in many ways he's quite legendary to our membership. So I'm really very honored to have received this prize, because this prize is only handed out at our IPCC meeting, so that's once in 3 years. This year, of course, it will be recognized in both Zoom and publication format. So I will, however, take for my publication a different topic than what I'm talking to you about today, so it's worth looking at both. Regulatory T cells are involved in both vitiligo and melanoma. I kind of already mentioned that, but in general, there is T cell involvement in either condition, and in one case that is quite undesirable. So if we look at vitiligo, and I will take my pointer here and kind of go there for you, we see here the characteristic symmetric lesions that develop in vitiligo skin. If we go down here, we see melanoma patient. You see, of course, where the tumor cells just have, the satellite lesions have really taken over in this patient after the primary tumor was removed. And you can see that the intricate relationship between these two conditions, where melanocytes are lost in vitiligo and melanoma cells take over in the tumor, that there is a very interesting relationship that is kind of mediated by the immune system. If you do have a strong immune response against your tumor, secondary to that response, you can develop vitiligo-like depigmentation. This is, for example, in a patient who received high-dose IL-2. And you'll notice that they also have very symmetric white lesions that develop in these patients. So here is an imagery that shows you the T cell infiltration as it can take place in vitiligo depigmenting skin. It also shows you where the CD4 and CD8 T cells are, because the red ones are your CD8s, the cytotoxic T cells. You'll see that the ones that are closest to the epidermis are actually the cytotoxic ones. And you can also see that there's a relatively larger proportion of those cytotoxic T cells in vitiligo skin if we compare this to melanoma lesions, as you can see here as an example. But I promised I was going to speak about regulatory T cells, and here this is important that we observed a paucity of regulatory T cells in vitiligo skin. Why is that important? Because if you think about it, these T cells that are mediating the disease, because they're killing off melanocytes, are not properly held in check by regulatory T cells, essentially because those regulatory T cells aren't there. And if we now compare lesional and perilesional and non-lesional skin in vitiligo patients, we really saw, very rarely would run into a regulatory T cell, whereas if you go up here to the control skin, you see about 40% of the cells are actually regulatory T cells because they have a red nucleus. So that's FOXP3 and blue is CD3. And you can also see here lentigo maligna, where there's several regulatory T cells involved, and the same holds true in melanoma, where this can actually be quite a nuisance because since regulatory T cells very handily readily make themselves available in these melanoma lesions and probably feel quite at home there, we really don't want them there because there they are impeding the exact responses that we want. So you'd like to inverse those situations if you could in some way. By the way, here's the quantification of the vitiligo skin. So you'd want to have more Tregs in vitiligo and less Tregs in melanoma. And that's kind of what I want to be talking about to you today. So we studied these conditions a lot using a model called H3TA2. I was involved in characterizing this model. It was developed by Drs. Nishimura and Mirotra. Where they built in a melanoma patient-derived T-cell receptor recognizing tyrosinase on the background of a mouse that is already transgenic for HLA-A2. And because the mouse will express both the majority of their T-cells recognizing melanocytes and the MHC class that's necessary to present the peptide to these T-cells, you can see that these mice will readily depigment over time. So they need both properties, both the HLA-A2 that you see here or the T-cell receptor that you see here, in order for this combination to actually take care of the melanocytes and mediate pigmentation. We can see here that because this is a very extraordinary T-cell receptor, it can function in the absence of any co-stimulation. So that means whether the T cells express CD4 or CD8, it really doesn't matter. The T cell receptor can respond to the HLA and peptide on its own without any help from CD4 or CD8. As you can see here in this picture, CD8 is helping this response. But that's not necessary. It'll function just by the TCR itself. That means that we can express this T cell receptor on CD4 or CD8 T cells to mediate the responses that take care of the vitiligo. And if you then want to, that's of course of importance for what we want to do here, because we think of regulatory T cells as being CD4 T cells. So we can use this mouse model to express the T cell receptor on CD4s and essentially try to induce regulatory T cells that can mediate immunosuppression. So the first way in which we thought we might try to overcome the depigmentation that naturally occurs and very rapidly in this H3TA2 mouse model is by introducing rapamycin. Now, rapamycin is something that can really help promote regulatory T cell development. And you can see here that if you take regulatory T cells, or if you take T cells in culture, and you then expose them to rapamycin, culture them in the presence of rapamycin, you'll see an increase in the percent regulatory T cells in your population. So we can do this also by taking T cells in total. Then from there, you might isolate your regulatory T cells, for example, based on their surface CD25 expression, pull them out, characterize their FOXP3 expression. If you're happy with the percent Tregs that you have, you can use this for adoptive T cell therapy. So this is what we did in our mice. Here you see the H3TA2 mice that were left untreated. Whereas if we treated them with rapamycin, and this is, as you can see here, for a period of 14 days, shown here, at 5 milligram per kilogram of body weight and delivered IP, then we see that we have a very good maintenance of the number of Tregs in the skin per surface area, shown here. We also see them in the circulation if you count that here. This is among splenocytes at end value. But what I think is most important is that you can see that rapamycin was really helpful in keeping the vitiligo in check. So rapamycin is one way in which we can introduce a greater proportion of Tregs in the mouse patients, if you will. We can also look at adoptive transfer. This is another way in which we could interfere with depigmentation. Here we see that we introduced really only 200,000 regulatory T cells into the animal's IV, and then we supported this treatment with IL-2 here. This is actually a relatively low dose, believe it or not, a little bit more than just low dose, which is actually thought to keep Tregs going. And we give this to them for 10 days, and then we follow them for quite a period of time to see when the vitiligo might or might not express itself. And as you can see here, the mice still look quite pigmented at the end of this treatment period. And at the same time, we also found that the number of regulatory T cells was still increased in the mice, and the same holds true if we look at the skin itself. You can see that happening here by double staining in the skin of CD3 and FOXP3. So this gives us two options. We can treat by rapamycin or by adoptive transfer, but there's another thing that we need to take into account. As I mentioned, for example, why we became interested in the homing pattern of these regulatory T cells is that in melanoma tumors, we find too many Tregs, and in vitiligo skin, we find too few. So we wanted to understand how that comes to be, and what we did then is to isolate, and this is in actual patients, the PBMCs, and then characterize their surface expression of chemokine receptors that would bring them to their respective tissues. We see that the number of regulatory T cells is not different really between vitiligo and healthy skin, for example, or with healthy PBMCs. Or between melanomas, maybe there's a trend, but there's nothing all that different about the numbers that are in the circulation. But then if we look at the surface expression of chemokine receptors that are important to bring these Tregs to their respective tissues, we didn't find much happening in vitiligo. So I should tell you that the homing patterns that are already imposed on the Tregs themselves are not different in vitiligo, but in melanoma, we did see that there was quite a difference. We see that the CCR4 expression especially goes up from about 45% to over 60%. So there's quite a few CCR4 expressing Tregs in the circulation. There's also an increase, like a doubling of just about of CCR8 expression, but we're talking about very small percentages there, and not much happening if we look at the percentage of cutaneous lymphocyte antigen expressed on these Tregs. But if we look at the MFIs, also here's an important difference also. So the cells that do express CCR4 also express a lot more of it. And for CCR8, that's also the case. Again, CLA is not the major driver for bringing these Tregs to the tumor. But CCR4 and to a small extent also CCR8 are overexpressed on Tregs in melanoma in the circulation. So this is what already drives their homing to the tumors. At the same time, if we look at the CD8 T cells in these same patients, we see that there's really no difference, or at least let's say there is much less expression of CCR4, like something like that's fourfold higher on Tregs than it is on CD8 T cells. We see that the expression of CCR8 is more or less equal among the CD8 T cells and the Tregs, and we see that the CLA expression is pretty much equal amongst CD8 T cells and Tregs, and that's given by these blue arrows here at the top. So that means that the CCR4 overexpression is quite unique to Tregs, and that it's different between patients and healthy individuals, which means that there's something to be done about this Treg homing to melanoma tumors. We see also that the skin-bound Tregs express CCR4. So if we isolate the Tregs from the tissue itself, that there is overexpression of CCR4 on these Tregs. And why do I say this? Because we want to make use of the fact that these Tregs can be skin bound as well. And so Another tissue that they really like to home to is the tumor. So if we look at the expression of some tumor-related markers such as TRP1, which is of course abundant in melanoma tumors, we see that the expression is here. And in serial sections, the CCR4 is found just right around there, actually in the same place where we also find FOXP3 expression. We also find CCR8 expression there. We find the chemokines that are responsible for attracting the Tregs, so CCL22 and CCL1. And we can see here, these are melanomas, but is one melanoma, but we see that if we look at lesions in other sites, so these are different patients where there were lung mets that we could isolate, we could look at lymph node and brain, that in every case there was abundant expression of CCL22 especially, which is the ligand for CCR4. We also know that we can use CCL22 to attract Tregs, and we see here that the number of CCL22 expressing cells are indeed much higher in melanoma than in control tissue of the same origin, and the same holds true for CCL1. So the chemokines are overexpressed, so are the Tregs, and that's probably in part because not only is the ligand overexpressed, but also the receptors in the circulation. And we know that this mediates migration because we did transwell migration assays where we add the chemokine to the bottom compartment, add the Tregs to the top compartment, and then we look at among the total number of T cells, actually, we look at the proportion that's Tregs in the top and the bottom compartment, and we see that this favors the migration of Tregs, especially in the case of CCL22 and not so much in the case of CCL1. So all of this points in the direction of CCL22, CCR4 as a very probable combination responsible for the Treg homing to the tumor, and also a possible mode or a possible pathway to intervene with this exact activity. And now if we go back to vitiligo for a moment, as I mentioned, there's very few Tregs in the skin, and actually we can look at the exact same cytokine as being an important factor in that case, because we evaluated the expression of CCL22 in the skin essentially by immunohistochemistry. We could see that there are cells that have very distinct expression of CCL22, and we could enumerate those in the skin. And you can see here that if you compare vitiligo to control tissues, that there is significantly less CCL22 expression in vitiligo skin. So if you remember, I mentioned that in vitiligo, the circulating Tregs did not have any less expression of the receptor for CCL22, but we do see a lot less expression of the ligand in the skin. And so again, here is an opportunity for treatment. So what we did then is to see if we could entice the Tregs to come back to the skin. And what we did is essentially cloning the CCL22 into an expression vector and then introducing this plasmid into the skin by means of a gene gun. We then see that we can have heightened expression of CCL22 expressing cells in the skin. That's shown here at the bottom and also quantified here at the top. And we can also see that this brings more Tregs into the skin, as shown here. So this is actually another graph where we looked at how much CCL22 would impact the differentiation of Tregs, and we see that that is not the factor we should be looking at. We know that TGF-beta can very much lead to differentiation towards Tregs. That's shown here in the lighter bars, and in the darker bars we see the differentiation in presence of CCL22, which essentially does very little for differentiation of Tregs. Sorry for that confusion. All right, so the introducing the CCL22 into the skin then can apparently very well introduce migration of these Tregs, because we could keep depigmentation at bay. And these are the same H3TA2 mice that I've been talking to you about. 3 weeks after vaccination, we see that there is much more depigmentation in mice that were not treated with CCL22 than those that were. And we can also see that that's accompanied by a much lower infiltration of the skin by our melanocyte reactive T cells, which are stained here by means of an antibody to the V-beta subunit of the T cell receptor. We also looked at the activity of these Tregs, and we see here that there is a very interesting connection between the interferon gamma producing spots in T cells that are isolated from mice that were not treated or treated with empty vector alone compared to those that were treated with CCL22. And so again, this would, these splenocytes themselves would also give us the same conclusion, namely that the CCL22 was able to overall reduce Treg activity in the mice after gene gun vaccination. So the principle that I just explained to you, namely that we introduce CCL22 overexpression into the skin to attract the Tregs there, as is indicated here in this little figure here on the right, this would bring the Tregs there based on the expression of CCR4 and CCL22 in the skin. And so we could actually use this exact same treatment to entice Tregs to go to the skin if they are found in melanoma patients. And why do we think that that would be helpful? Because we know that in melanoma, there's an overexpression of CCR4. They might even be more enticed to go to a place where you have a lot of CCL22 expression. And we know that they have a tendency to go to the tumor, which is exactly where we don't want them. So if we can just reroute them and send them away from the tumor and instead have them go to the skin where we've overexpressed the CCL22 even beyond what we find in the tumor, that would then, we at least propose that that would lead to less tumor infiltration in the sites where we would have introduced it. So if we introduce our tumor cells IV, they will home to the lung, and we can quantify the number of tumors that establish themselves in the lung. And we did this for both mice that have been exposed to empty vector to the skin or CCL22 to the skin. And you will appreciate here that the number of tumors in response to mouse CCL22 was much less. And that was pretty interesting, right? So at that point, just because we had attracted the Tregs to the skin, we could impact the efficacy by which the actual effector T cells were trying to keep these tumors in check. We also tried this in a therapeutic setting. That's, as you might know, quite a bit more challenging, but fortunately we could also show here that once we had the tumors already set up in the mice, we could still have an impact on the growth and establishment of new tumors in these mice by comparing the tumor number and the tumor area that was occupied by tumors in response to skin vaccination with CCL22. At that point, we also find that the treatment leads to a greater proportion of Tregs in the skin. So here we see skin treated with empty vector DNA, and here we see skin that's been treated with CCL22 DNA. And you can appreciate that the number of Tregs is much greater in response to CCL22. At the same time, of course, what we then want in the case of tumors is that less of these Tregs would be found inside the melanomas so that we can show that the mechanism is indeed what we think it should be. Here we see in untreated tumors, the Tregs, and this is just to show you how we quantified that. So obviously we don't want as many of those Tregs in the treated, in tumors from treated mice, but here's FOXP3 expression, here's CD3 expression, and here would be an example of Tregs. And if we now compare those numbers for empty vector and CCL22 treated mice, we do see a slight reduction in the number of Tregs. Now that sounds like it's just a minor difference, but the importance is, of course, how that impacts the T cells and perhaps the NK cells that might be suppressed by Tregs. And we see here that it does really help for the number of T cells that we can find in these cases, and also to some extent the number of NK cells that we find in the tumor. So it's more about the proportion between Tregs and T cells that we should count on as this being a mechanism to divert Tregs and at the same time support anti-tumor responses. So some future directions that we'd like to take with this mode of work, or with this line of work, is that we'd like to see what we could do with redirecting Tregs in the case of checkpoint inhibitor-induced skin rash, where we see, of course, a big inflammatory response that really requires some extra Tregs to take care of it. And why we think that's important is because skin rash itself may not be considered the most important adverse event that can happen after checkpoint inhibitor applications. But we do know that it is the earliest adverse event that tends to take place in patients. So we think if we can overrule this type of adverse event early on and still at the same time, bring these Tregs to the skin and bring them away from the tumor, that we have a double whammy in supporting anti-tumor responses. And another thing that we want to take from here is to provide even greater specificity to our Treg therapies by adding antigen recognition to these Tregs, and this is particularly important for vitiligo, and we hope to present some data to you about this very soon. So essentially it helps for the Tregs to be activated only where the expression of a relevant antigen is found, so preferably in this case in vitiligo perilesional skin. So if I can summarize this, we looked at interferon gamma expressing T cells that mediate vitiligo and melanoma, but at the same time the control by Tregs is diminished in vitiligo and is exaggerated in melanoma. And in vitiligo, indeed, we find less of them in the skin, but they are very abundant in tumors. So though we have autoimmune responses that are inhibited by adoptive Treg transfer, we can also introduce antigen-specific Treg to add specificity. We can think of rapamycin treatment to enhance Treg numbers, leaving reduced depigmentation, and we can also count on the overexpression of CCL22 to bring Tregs to the tumors, which we can divert by overexpressing the CCL22 in the skin, and making use of the CCR4 overexpression on Tregs in the circulation. If we overexpress CCL22, this at the same time can overrule depigmentation that we find, and we hope that the same would be true in actual patients. And the general conclusion that I'd like to leave you with is that redirecting Tregs can enhance anti-tumor responses and control autoimmunity. So this is the has been a holy grail for us, at least, to see if we can do something for melanoma and vitiligo in one go. And this might be a way to do that. So I've been very fortunate with a very exciting group of investigators that have helped me over the years. And this is just a picture of last year, but I do want to make a special mention of two people that have been very involved in our Treg work. So in my lab, that's been Jusip Mukatayev, who is graduating on this topic at around this time, of course, very much encountered difficulties because of COVID-19. And then Shikhar Mehrotra, a lot of the work that I presented today was from a grant that we held on Tregs, and we did that work in very well thought out collaboration. So I'm very thankful for that period. Okay, if you have any questions, please don't hesitate to contact me. I shouldn't be hard to find on the IPCC website. And it's been exciting to present this to you today. Thank you very much.